Hey guys, so I'm Sarah Driver, and I'm going to be sharing you guys a step of our procedural process when we're doing our project. So my partner is Miranda Murphy, and our research project is a QTL analysis study looking at type 2 diabetes and inflammation. So let's get started. Alright, so step 1 of a QTL analysis is to run genome scans. So to do that, I have my script here, and I load the library, the QTL library. In QTL, it's a package, so you're just loading the things that are in that package. Then I load my B2BR data. I already did this, but it's just in my downloads, and I go like this, and I press load, and yes. And then I define some functions that I'll need later. And then I would locate the gene that I picked out, and I'm using CCL5, which is the RANS gene. It's involved in chemokine signaling because our research project is looking at phenotypical traits associated with type 2 diabetes and inflammation-related genes. And to find out what you have to call it in RStudio, which is the software we're working with, we use a mouse gene database, and here you see it's RANS, but it's called CCL5. So this is what it's called in RStudio. So you do have to do that after you figure out a gene, and then figure out the ID number using these two. That's just helping us get it out of the data. We retrieve expression in adipose because we're looking at adipose tissue right now. And moving everything we want to look at into the cross object. Like I said, we're looking at type 2 diabetic traits, so here we're looking at glucose and insulin and adipose weight. Look at pairwise plots. This is the pairwise plot. So looking at it, we look at this column only because we don't want to know why the phenotypes are related to each other. We just want to know why they're related, or if they're related to the gene. And it's showing correlation values. So the strongest one seems to be here at insulin at 10 weeks. But these just kind of give you a clue of what's going on. So now we're going to pull out sex as covariate and run our first scan. Alright, so I've actually already run this permutation test, so I'm not going to run it again because it takes forever. But that just identifies a threshold that it puts on your graphs. So you can tell, you can use it as a basis for what's significant and what you want to look at. Because it generates these log score plots, and that stands for logarithm of odds score. It's basically plotting the probability that the gene that we're looking at and the phenotype are related. So a high peak indicates a high probability. So like here on chromosome 16, that's a good sign. That means that this gene right here, something's going on chromosome 16. So that's the one we want to look at, and I want to see if that is replicated in any of the other log score plots with the phenotypes. Okay, so here we go. Here's one. See, at glucose at 8 weeks, we have a peak here, shared. That's good. Not really anymore. And then we do summary, because you want to see if the confidence intervals overlap. And the confidence intervals are intervals on DNA. They're in centimorgans, and they tell you roughly where the peak is occurring. So if they overlap, then that means the peak is occurring at relatively the same place, and that's a good sign. Okay, so here, this is chromosome 16. At glucose 8, we have a low of 2, high of 41, and here we have a 20 to 31. So this 20 to 31 does fall between 2 and 41, so that's a good sign. And then the next step would be conditional scans with this. But this is step 1, so that's all I really wanted to show you. We also looked at triglyceride levels as well when we were doing the genome scans, but it's basically the same thing except for where I put insulin and glucose into the cross object, I put triglycerides. So